All right, so this is the part of this chapter that people usually have the biggest issues with. And it's using the words hypertonic, hypotonic, and isotonic. So that end of the word tonic has to do with solute concentration. And hyper means really high concentration. Hypo means very low. And iso means equal. So those are the three words that we're going to be using. So if we talk about a cell sitting in a solution, there's basically three ways that it can look, right? You can have this situation where you've got a cell sitting in a very concentrated solution. So the solution is more concentrated than the cell. Now, all those dots are going to be solutes. Let's just say this is salt, okay? So there's more salt outside the cell, which means there's more water inside the cell. So hopefully that makes sense. Now, if we look at this one, the cell has more salt in it, and the solution has more water in it, okay? And then this one, you can see that they're equal. Okay, so now we're going to put words to that stuff. So we're talking about the solution here that the cell is sitting in. So if we look at this one here, the solution has more solutes than the cell. So if we're talking using those words, we would say this first one is hypertonic. Okay? Now the second one is going to be the opposite. So we're going to call it hypotonic. Okay, so you've got hypertonic in this first one because the solution has more solutes than the cell. Hypotonic in this one because the solution has less solutes than the cell. And then this one where it's equal is isotonic. Isotonic. Okay? So that's going to be really important because this is going to tell us what direction water is going to flow. So let's look at this again. Woo! I'm erasing stuff, aren't I? Okay. Sorry. Okay, so we've got our hypertonic solution that the cell is sitting in. So where there's blank space is water. What direction do you think water is going to travel in this situation? Is it going to go into the cell or out of the cell? So hopefully you're going to say water is going to move out of the cell because there's less water outside the cell than inside the cell. So the cell is going to lose water in that situation. In this next one, you've got a lot of water outside the cell, not a lot of water inside the cell. So to go from high to low, the water is going to go from outside into the cell. So water is going to enter the cell. In this last one, where it's equal, there's just going to kind of be a free flow into and out all the time, but no real net movement of water. Okay? So depending on whether you have an animal cell or a plant cell, different things are going to happen. So if you have an animal cell, that does not have a cell wall. So what's going to happen is, in this situation, a hypertonic solution, what's going to happen to an animal cell is we said the water is going to exit the cell. So the cell is actually going to shrivel up and do something called crenating. And I have a picture of that somewhere in here. Somewhere. There it is. Okay. So water going out, it's going to shrivel up the cell. Now, in this next situation, hypotonic, what's going to happen is water is going to go into the cell. Well, it's just going to keep going into the cell until eventually the cell explodes, and that's this situation here, and that's called lysing. Then, in this situation, this is when an animal cell is happy because it's in an isotonic solution. Okay? Now, plant cells are going to be different. Plant cells, in this situation where water exits the cell, it's going to do something called plasmolysis, which is this right here. And plasmolysis is going to be where water exits the cell, and the cell membrane is actually going to pull away from the cell wall. Not a good thing, okay? Now, if you put a plant cell in an isotonic solution, it's not happy there either. It's going to be it's alive, but it's kind of flopped over. So like if you haven't watered your plants in a while, that's how a lot of mine look. And then you've got turgid. This is going to be how a plant cell likes to be. And that's going to be when it's in a hypotonic situation. And so what happens is it's going to take in water, right? Water's going to go in. And what's going to happen is it's going to cause the membrane to push out against that cell wall. And that's what makes a plant able to kind of stand up. So those are going to be the three ways that a plant cell can look in response to those situations. Okay, so this stuff is really important because let's say that you're talking about giving someone an IV, right? Our cells aren't pure water. There's a little bit of salt in it. So that's why they give you that saline solution when they give you an IV. If you gave someone a water, purely water IV solution, what do you think would happen to their blood cells and their other cells? Well, if we think about it, you're going to create a hypotonic solution, right? You're going to dilute that solution. And so there's going to be more water outside the cell than inside the cell. So it eventually would make your patient's cells pop. That's why it's so important to understand those terms and to understand why water moves in what direction. Okay. Now, as far as other types of cells, they can have controls over this stuff too. Protists, so those ones that have that contractile vacuole, they can control their water intake that way. So if they're taking in too much water, they could just pump it out through that contractile vacuole. And that's how they can kind of maintain themselves. Now, the last thing that we're going to get into here is going to be moving things into and out of a cell. And I actually have a very terrible animation that I forgot to download here. Let's see if I can get it. And so this is going to be what's called exocytosis and endocytosis. And this is a really old, decrepit type of animation. But anyway, this is how we can like move large amounts and large quantities of things into a cell. Let's see, it doesn't seem to be working. Oh, here we go. Open with, I'm going to use, actually, I should just use, let's try QuickTime Player. And so what's going to happen with endo and exocytosis is you're going to be able to take bulk movement of water. Here we go. Yay! All right. So this is going to be a picture of exocytosis. So there you can see what just happened, right? So if I go backwards. The way I remember exocytosis is something exiting the cell. Okay, oh, now everything's coming up now. That's awesome. Okay, so what's going to happen is you've got this vacuole of food that you want to get out of the cell. So what your cell's going to do is that vacuole is going to get close to the membrane, and then it's going to eventually fuse with the membrane, and then it's going to dump its contents out. That is exocytosis, stuff exiting the cell. If we were to go in the opposite direction, that would be endocytosis, and that would be stuff coming into the cell. So if you look here, I'm just going to play this backwards. You can see we want to take stuff into the cell membrane's going to kind of fold in, and then eventually close off, and now that is inside the cell. Okay, so exocytosis, endocytosis, exo, endo. Okay, so that's the difference between the two. Now, of course, there's going to be some specialized forms of that. And we've got phagocytosis, and phagocytosis is going to be where you have a membrane that's going to surround something and then engulf it. And then you also have penocytosis, which is taking in a large amount of water. Now, when would this stuff happen? Well, phagocytosis is something that an amoeba does when it's feeding. But I'm going to show you a video of how your white blood cells do this. And this is going to later when we talk about how this stuff works. And it's a pretty cool video actually showing this stuff happening. So white blood cell eating bacteria. There we go. It's a really cool way to show you what phagocytosis actually looks like. And this is why we have our white blood cells. So this is really just a neat thing to kind of get you to remember how this stuff works, I think. So hopefully this will work. Yes. Oh, this is such a cool thing. I just think it's so awesome to see your body actually doing stuff. So those little things you see there are going to be bacteria, and this thing that's like a blob moving is a white blood cell. So what it's actually doing is it's chasing after that bacteria, and it's going to try and engulf it. And eventually what's going to happen is it will find that bacteria, and it's going to phagocytose do phagocytosis. See, there it just did it. It just kind of engulfed it, right? So that's exactly that happening in our bodies. And that's a great thing because that's what keeps us from getting super sick and that type of thing. So that's going to be how we are going to move stuff into and out of a cell. And in the next chapter, we're going to start getting into energy and how that all is going to come into play.